Hello, I'm Maria Williams with the Collaborative on Health and the Environment. Change was bringing you the latest environmental health science through our partnership calls and webinars, science serves, publications, and social media. I would like to welcome everyone to today's CHE EDC Strategies Partnership Webinar, which is titled Preconception Environmental Exposures and Children's Health. Our moderator today is Carol Kwiatkowski, Executive Director of the Endocrine Disruption Exchange. We will leave time following the presentation for a brief Q&A session. You may type in questions through the Q&A feature available on the menu bar at the top of your window at any point during the presentation. After the presentation, our moderator will read out questions for our speaker to respond to. We will get to as many comments and questions as we can during the Q&A period. For those of you who called in on the phone, we have posted slides to accompany today's webinar on our website. You can download these by going to healthandenvironment.org. Please scroll to the bottom of the page and select today's webinar. On the webinar page is a link to the slides. Everyone on the webinar right now is muted with the exception of our moderator and our speaker. This webinar is scheduled to last for 30 minutes and is being recorded for our call and webinar archive. With that, I'll turn things over to you, Carol.
Thank you, Maria. Welcome, everyone. We're very honored today to have Dr. Joseph Braun presenting for us. He is an assistant professor in the Department of Epidemiology at the Braun University School of Public Health. He will be reviewing the human, animal, and mechanistic research on preconception, environmental exposures, and children's health. Most interesting, in my opinion, is research on how environmental exposure in the father contributes to health outcomes in the children. We'll be learning a lot about that today. We will also get to hear recent findings from the PEACE study, in which Dr. Braun is investigating the role of preconception exposures in children's neurodevelopment and cardiometabolic health. Dr. Braun was formerly a school nurse in Milwaukee, Wisconsin, before receiving his master's and doctoral degrees in epidemiology from the University of North Carolina at Chapel Hill. He studies the patterns, determinants, and health consequences of early life environmental chemical exposures in pregnant women and children from the United States, Canada, and Mexico. He has special interest in studying obesity and neurodevelopmental disorders like ADHD and autism, and is particularly interested in modifiable sources of environmental exposures. 3 cheers for that. So thank you, Dr. Braun, for being with us, and the floor is yours.
Thank you so much, Maria and Carol, for inviting me here to talk today. It's my pleasure. All right. So before I begin, I just want to say that I have no financial conflicts of interest related to the material being presented. However, I was financially compensated as an expert witness in a lawsuit related to secondhand tobacco smoke exposures. So Homer Simpson, the father of the US television show The Simpsons, once said, "I never thought of fatherhood as something that could affect a kid." And I think this quote aptly summarizes our current state of knowledge about how paternal exposures and experiences before the conception of a child influence their health and well-being later in life. And so today I'm going to talk to you about how both fathers and mothers' exposures before they become pregnant could influence children's health later on down the road. So there's new results that I'm going to talk about here today that are challenging 2 paradigms that we've held as key assumptions in the study of environmental chemicals and children's health. The first is that gestation, infancy, and childhood are the most important periods of susceptibility to environmental exposures. And the second is that fathers can only influence child health through Mendelian inheritance, i.e. genetics. Now, there's been justifiable emphasis on studying the impact of a wide array of environmental exposures on the fetus and child and relating those exposures to later life child health. This includes things like air pollution, pharmaceuticals, toxic chemicals, as well as our diet. What hasn't been done until recently, that I'm going to talk about in more detail today, is how some of these same exposures can influence the child via maternal and paternal transmission through the gametes, and specifically when these exposures occur before conception. So this opens up to a new window of susceptibility that we need to be thinking about when we're examining the impact that these environmental exposures might have on human health. So one of the more recent papers on this that has prompted a lot of research in this area is a paper by Diaz and Ressler on paternal fear conditioning. And in a series of experiments they did with rodents, they made male rodents afraid of the scent of acetophenone by presenting the scent to them and then shocking their foot at the same time. They then mated these fathers to females and looked at whether or not their offspring in the first and second generation were also afraid of acetophenone. And they found that actually these offspring had enhanced startle responses to the scent of acetophenone despite never having been exposed to it before. What was even more compelling was that there were neuroanatomical changes in the olfactory bulb of these offspring that were specific to acetophenone. And they also observed DNA hypomethylation in several of the genes related to this pathway. The results became even more compelling in my mind in that they did a series of experiments where they were able to recapitulate some of these phenotypes in both offspring that were conceived via in vitro fertilization. So the father never actually came into physical proximity with the mother in the case that there was some sort of reproductive behavior that he was engaging in that might have affected the offspring. And they also did cross-fostering experiments to show that these effects were robust there as well. In two papers by Rogers et al., published in 2013 and 2015 from Tracy Bales' group, they found that additional evidence that paternal exposures before conception could influence offspring health. In this case, they used a stress paradigm where the fathers were subjected to some fairly extreme stress, and they found that offspring conceived from these fathers had decreased hypothalamic pituitary adrenal axis responses, as well as changes in sperm microRNAs in the fathers. In the second paper that they published, they were actually able to recapitulate this offspring phenotype with specific sperm microRNAs that they identified in the first paper. So they actually took a set of these sperm microRNAs and injected them into an unexposed zygote and were able to recapitulate those phenotypes. So these two papers have prompted additional research looking at how environmental chemicals in rodent studies might impact neurobehavior. And this is a very recent paper by Fan et al. From PLOS One, where they were exposing male rats to bisphenol A at a relatively high dose of 50 micrograms per kilogram per day. And they looked at how whether the offspring born to these fathers that were exposed to BPA had increased anxiety in both an open field test and an elevated plus maze, as well as their corticosterone levels both before and after the test. What they found was that the females born to these fathers had more signs of anxiety and on these two neurobehavioral tests, as well as higher post-test corticosterone levels. So this suggests there could be some sex-specific effect of BPA on later life neurobehavior in the offspring. Another study by Harris et al. From Emily Rissman's group, which was published recently this year, looked at male mice that were exposed to BPA and did not observe an effect of paternal BPA on offspring anxiety or offspring vocalizations. They did find that these males who were exposed to BPA did weigh less than the control group, and they also found, so this result suggests there could be some species-specific effects of paternal BPA exposure on offspring neurobehavior, given that the prior study by Fan used rats and this study used mice. But these are the two of the studies that we have to date on this. Now, what about epidemiological studies? What do we know about in humans? Now, most studies looking at preconception exposures have been limited to looking at outcomes like childhood cancer, birth defects, as well as some birth outcomes. And most of these relate to occupational exposures experienced by the mother or father before conception. And in two meta-analyses or systematic reviews, paternal benzene and pesticide exposures before conception have been linked to childhood leukemia. And there's also been a number of studies linking different pollutants, things like including metals, PCBs, as well as some solvents, to a variety of offspring health outcomes like birth weight, as well as some childhood cancers and birth defects. But there haven't been any detailed studies looking at specific environmental chemical exposures that have been measured with newer state-of-the-art biomarkers and relating these to later life health outcomes in children. One of the more recent studies on this that has been done to look at these potential transgenerational effects of exposure looked at the effect of DES on ADHD risk. And in a paper published in JAMA Pediatrics by Marianthi Kumarzoglu, she found that grandchildren born to grandmothers exposed to who used DES during their pregnancy were at increased risk of ADHD. In fact, they were 36% more likely to have ADHD compared to grandchildren born to women who didn't use DES. This effect seemed to be the strongest for first trimester DES use, where the odds ratio was 1.6, and they didn't observe any sex-specific effects. What's really interesting about this study is we know that DES is a very potent endocrine disrupting chemical, and this data suggests that it could have an effect on the developing oocytes among these grandchildren while there are oocytes in their mother's belly, while their mother is a fetus. And this opens up the door for the potential for other endocrine disrupting chemicals to have an impact on offspring health. We've been looking at this with regard to phthalates, which are a class of endocrine disrupting chemicals that have several well-known health effects in both animals and humans. And phthalates are this non-persistent multifunctional class of chemicals that are used in food packaging and processing. They're used to make polyvinyl chloride plastics softer. They're used in some paints and adhesives and floor backing, as well as some things like rainwear. And there's been concern over their toxicity given the well-documented anti-androgenic effects of phthalates in both animals and humans, as well as evidence of thyroid disruption and their ability to interact with the peroxisome proliferator activated receptor. A recent study by Howard Wu from Rick Pilsner's group found that men who had higher exposure to some phthalates had alterations in their sperm DNA methylation that were specific to genes related to growth, development, and cell movement and structure. So this suggests that phthalates could have an effect on the on this DNA methylation that could be passed on to the offspring. What isn't clear is whether or not these changes in DNA methylation are linked to later life health outcomes in the offspring conceived from those sperm. So we've been investigating these questions using a new study called the PEACE study, or the Preconception Environment and Childhood Health study. This is a study that's nested within the Environment and Reproductive Health study, or EARTH study, which is a preconception cohort started by Russ Hauser back in 2004. And this cohort's been enrolling subfertile couples from the Massachusetts General Hospital Clinic in Boston, Massachusetts from 2004 onward. We have detailed measures of maternal preconception and prenatal exposure, as well as paternal preconception exposure to phthalates and phenols, and also have banked biospecimens that would allow us to look at things like persistent pollutants like perfluoroalkyl substances or flame retardants. We just recently received funding from NIEHS to do follow-up of these children at age 6 to 10 years of age, of whom there's about 550 children who have been born in the cohort, and we're hoping to bring about 300 of these children back in over the next 4 years. We're assessing an array of health outcomes, including neurodevelopment, asthma and allergy, pubertal development, body composition, and cardiometabolic risk. So this is a schematic of the study that we're running, where we're in the original EARTH study. Women were enrolled along with their partners, and we collected blood, urine, and hair, sperm from a subset of men, follicular fluid from women who were receiving IVF. We have a detailed exposure history that is administered at baseline, and then additional exposure questionnaires throughout follow-up. We measured diet with a food frequency questionnaire, and then collect information about socioeconomic status. During pregnancy, we collect blood and urine 3 times at each trimester during pregnancy, and also have hair samples from these women. And we also collect a detailed exposure history and information about labor and delivery. The new follow-up visits, which we just began in June and have completed about 12 of now, are collecting blood, urine, and hair from children, and we're measuring children's IQ, behavior, executive function, body composition, cardiometabolic markers, as well as eating behaviors. These are some preliminary data that we published that were the basis for the grant that we just received, where we've been looking at the relationship between maternal preconception urinary phthalate concentrations and externalizing behaviors in these children when they were 2 to 7 years of age. Externalizing behaviors are things like hyperactivity and aggression, and are often thought to be and are often elevated in children who have ADHD, specifically at the hyperactive type. By and large, we saw mostly null associations between these phthalates and parent-reported externalizing behaviors. However, we did see some evidence that increasing urinary monoisobutyl phthalate concentrations were associated with more externalizing behaviors in boys, but not girls. And these beta coefficients here are such that a positive beta coefficient indicates that urinary phthalate concentrations are associated with more of the behavior in the child. So in this case, there was a sex-specific effect for monoisobutyl phthalate. These are data showing the relationship between the paternal preconception urinary phthalate concentrations and internalizing behaviors now. So here we're looking at a slightly smaller subset of children because we don't have data from all of the fathers, and we're looking at behaviors related to anxiety and depression, which are these internalizing behaviors. Again, positive coefficients indicate that the phthalates are associated with more of that behavior. In this case, we see a much different pattern of results where we see that increasing phthalate concentrations in some cases like DEHP are associated with reduced internalizing behaviors. In other cases, like mono-N-butyl phthalate, monoisobutyl phthalate, and monobenzyl phthalate, we see that increasing concentrations are associated with more internalizing behaviors in the boys and null or even protective associations in the girls. And this modification by sex is statistically significant in some cases. For the other two phthalates, we did not see any evidence of an association between paternal phthalate levels and child internalizing behaviors. We've been doing some additional work looking at preconception exposures and birth weight in this cohort. Carmen Masurlian published a paper reporting that paternal diethylhexylphthalate metabolites measured in the urine were associated with lower birth weight in 195 father-infant pairs from the EARTH study. And this association was actually stronger among babies who were conceived via IVF, suggesting that maybe that there's a potentially sensitive subgroup among couples who are receiving fertility treatments. And we saw that in this study that each log unit increase in DEHP metabolite concentrations was associated with a 90-gram decrease in birth weight. These associations in this study actually became stronger after we adjusted for maternal prenatal levels of DEHP. So this association was independent. This association between the father's exposure and the infant outcome was independent of what the mother experienced during her pregnancy. In another paper published this last year, Mustielis et al. Used the EARTH study again and found that maternal preconception BPA exposure was associated with reduced birth weight, where each log unit increase in urinary BPA concentrations was associated with about 120-gram decrease in birth weight. And again, this association remained even after adjusting for prenatal exposure to BPA. This is some work in progress by Julie Scara, who's a master's student working with me, where we've been using the EARTH study data to look at preconception urinary triclosan concentrations and thyroid hormones among women in the cohort here at baseline. And in this cross-sectional study, we found that higher triclosan levels were associated with reduced free triiodothyronine concentrations measured at that same visit. Triclosan is an antimicrobial chemical that's used in a wide variety of consumer products, most notably in a specific toothpaste as an anti-gingival agent or an anti-plaque agent. And it's also used in some toys, fabrics, and other household goods, and was recently banned by the FDA from over-the-counter hand soaps and sanitizers because of concerns of developmental toxicity and antimicrobial resistance. And here in these data, we're seeing, though, that triclosan exposures before pregnancy may impact thyroid function in these women. And now what we're going to do next is determine whether or not triclosan and some of these other phenols are related to child behavior that we previously mentioned, previously measured as part of our pilot studies for this larger piece study. So there's some challenges to conducting epidemiological studies of preconception exposures and some potential solutions. One of the real big challenges is enrolling couples at risk of becoming pregnant. About 10% of US women become pregnant each year in the United States, and about 2/3 of these are intended pregnancies. So there's a relatively small pool of women who are trying to get pregnant at any time, and thus it is challenging to find them and recruit them into your study. Some solutions to this are to use fertility clinic enrollments like we have been doing in the EARTH study and our new PEACE study. Other methods include targeted internet advertising through Facebook or Twitter. This has been done by Lauren Wise and Elizabeth Hatch in their Presto cohort, where they're enrolling women into an internet-based cohort. And then others have taken more of a brute force approach to just identify large rosters of people and contact them and determine whether or not they're planning to get pregnant anytime soon. And this was done by Jermaine Buck-Lewis in the LIFE study. There's also the potential for loss to follow-up and censoring at multiple time points during the course of follow-up in doing these studies. And this censoring and loss to follow-up can both reduce sample size and introduce selection bias into our epidemiological studies. So we have to be thinking about the fact that women, there's a probability of of conceiving and being in the state of pregnancy, and then conditional upon that, there's another probability of having a live birth, and conditional upon that, there's the probability of continued follow-up in a study. So this requires that we have large sample sizes to account for all of the places in which we can have loss to follow-up, as well as looking at whether or not there is any sort of potential selection bias being introduced by informative censoring or loss to follow-up. In addition, another solution to this is to do retrospective assessment of some exposures. And this was done in the paper I mentioned earlier about DES and ADHD, where they used the large nurses' health study and actually used retrospective assessment of the DES, where the mothers were recalling whether or not their mothers had used DES during their pregnancy. Finally, there's always concerns about generalizability when we're working with groups of people who are trying to get pregnant or who attend fertility clinics. But I would argue that one of the more important things first is to make sure that we have a scientifically valid study. And if we have a scientifically valid study, then we have good results with good internal validity. The generalizability can follow as we do more studies and replicate things, but to start, it's important to have this internal validity first before we start worrying about the generalizability of the findings. So I just want to conclude here by saying that there's now evidence and a growing body of evidence to suggest a potential for preconception environmental exposures to affect children's health, and that in some cases it looks like fathers' exposures and experiences before conception are determinants of later life offspring health. And this has important public health implications as we think about how we do risk assessment for environmental chemical exposures, in that there's now a new susceptible subgroup we have to be thinking about. We've had justifiable emphasis on thinking about the prenatal period or infancy or childhood, but now we need to be thinking about couples who might become pregnant and whether or not their exposures are important to later life health outcomes. It's also important to be thinking about this from a clinical perspective, in that practitioners who are counseling patients who are about to become pregnant or trying to become pregnant might need to be thinking about what sort of exposures and experiences they're having and how those might affect offspring health, in addition to having a healthy pregnancy. So I'd want to conclude by acknowledging colleagues and collaborators at Braun University, Harvard School of Public Health, U.S. Centers for Disease Control, as well as funding from the National Institutes of Environmental Health Sciences. And thank you for listening.
Thank you, Joe. That was a great presentation. I've really enjoyed that. And the pictures at the end are wonderful. Your children, in case people can't recognize that. Well, I'll kick off with a question that is a little bit off topic, but are you aware of research on differential exposures between boys and girls to these sorts of chemicals, possibly dependent on the toys they play with or personal care products they use in young children?
That's a good question. So we've looked at patterns of exposure, and in some cases we see we don't see sex as a strong predictor of a lot of these exposures. I mean, exceptions include things like monoethyl phthalate, which is the metabolite of one of the phthalates used in personal care products, tends to be higher in women than men. But that sort of difference doesn't emerge until later on when girls start engaging in more personal care product use. The other thing is that some of these exposures differ because, or levels of the biomarkers differ because of inherent differences in the biology of men and women. So for instance, some of the persistent pollutants are higher in men compared to women because women lose blood through menses, and that menses is a way for them to excrete some of those pollutants. They also, if they've been pregnant, they've offloaded some of the pollutants onto their feet, into their infant, unfortunately, and also do so through breastfeeding. So, but in terms of specific behaviors, it's possible, but I'm not aware of anything related to toys.
Okay, great, thank you. So we have a listener with a question. She says, do your studies differentiate demographics and cultural diversity as a component?
So we have adjusted in our analyses for some basic socio-demographic factors, including things like maternal education, age, marital status. We also looked at the parents' ADHD-like behaviors or externalizing behaviors in internalizing behaviors. What we haven't done, what we're doing now in these newer studies is collecting more detailed information about some of these things. What is nice about the study we're using is that it's a relatively homogeneous population with respect to race and socioeconomic status. These are relatively high income and well-educated couples from the greater Boston area who are seeking these fertility treatments. So that reduces some of that variability, but not all of it.
Great, that's good to know. Have you done any analysis of combinations or multiple compound exposures?
That's the next step. That's what we're hoping to do with this new funding we got and with the new follow-up, is to look at the potential effects of mixtures of phthalates as well as mixtures of other chemical exposures.
Mm-hmm. Okay. Do you know of relationships between the externalizing behaviors and the internalizing behaviors for those studied in your research?
I don't know about the correlation in this study off the top of my head, to tell you the truth. Sometimes they are correlated. Some of these behaviors are related to each other, particularly later on in adolescence. Children with ADHD sometimes have some depression because their behaviors can be a little bit alienating for them socially. So these disorders, but these disorders can also be related because of sort of shared common biological mechanisms that affect neurodevelopment or behavior.
Great. Are there studies on the children or grandchildren of Vietnam vets exposed to Agent Orange?
Oh, my colleague down the hall would know this because he's done all this work with the Agent Orange people. I would look to see. I think there's been efforts to do this, and actually I think there's some interest in the Department of Defense right now to do this. And there should be a report coming out soon on this from the national academies. And so there's interest in doing this. I think there's been some crude analyses of it in terms of like things that they've looked at, but nothing real comprehensive. I think there's been some record linkages, but I don't think it's specific to Vietnam vets. I think there's some stuff related to the first Gulf War, though.
Okay, great. I'm going to combine two questions now. There's one from a person who showed up a little late and asked if you could clarify on the hypothesis of why men's preconception exposures matter. And then the other question is there enough data on changes in preconception lifestyle, for example, exposure periods for each parent, dietary changes, etc., to support differential analysis for benefits of such changes and how they might be effective?
So the first question was with regard to the potential mechanism here is that, yes, so there is evidence from these animal studies that some of these non-chemical exposures can alter both sperm DNA methylation as well as microRNAs that are present in the seminal fluid, and that this may be the way that some of these paternal exposures are being transmitted to the offspring. And it makes sense evolutionarily that the father would need to contribute some information about what the environment is like to his offspring, and this needs to be a malleable mechanism, and epigenetics are a good way to do that. With regard to the exposure variability, so there have been some studies with regard to the diet changes from the overholics cohort in Finland, I believe it is, as well as the group using the allspac cohort. And so there's a paper by Pembry that's looked at this, as well as some others, that has looked at differences in food availability and offspring health. With regard to variability, things like phthalates and phenols are good candidates to look at with regard to variability because people's exposure changes due to their behavior and lifestyle. So this is part of the reason why we selected phthalates, is we can look at whether or not exposures that are higher during certain periods of spermatogenesis or at certain periods relative to the IVF cycle among those getting IVF are more important than exposures at other time points.
Mm-hmm. Okay. Have you observed any associations between sperm quality and child behavior?
We have not done that. We could do that with these data. So thank you for the idea. But I don't know about if there's a literature on this. I'd be very curious if there is. So if you're aware of anything, so please send it to me. But I think it's a good question to look at.
Great. And were the children of the grandmothers exposed to DES from sons or daughters of DES mothers? The grandchildren, sorry, of the grandmothers.
The grandchildren were both boys and girls. This is in the nurses' health study. So all of the mothers are women, and they asked those mothers about whether or not their mother, i.e. the grandmother, took DES during her pregnancy. And then they have follow-up on the children of the mothers who were enrolled in the nurses' health study.
So they were the daughters of DES mothers that they were interviewing?
Yes, daughters of and non-DES mothers.
Okay, great. Let's see, we might have to end with this one, although we still have a couple coming in. But someone asked, do you have a basic list of things to avoid for both mother and father preconception?
You know, I think a basic list, no. I think, you know, there's a podcast I just did with Sophia Goucher that's posted on her website. I think it's Tips for Non-Toxic Living. I cannot remember, but her name is Sophia Goucher, Gushee, and I give some information there about potential ways to reduce your exposure. There's also information from the pediatric academic societies, as well as the Academic Pediatric Association environmental health groups that have put together some sheets. They might be a little dated now, but there is information out there on the web that you can find about how to reduce exposure to some chemicals.
Terrific. Well, I think that's a great place to end. And I want to thank you again for your great presentation and for answering all of the many questions. And thanks to everyone for participating.
Thank you.
Great. Thank you so much, Carol. We are approaching the end of today's webinar. A video recording will be available on the CHE website soon, and tomorrow you'll receive an email containing a link to the recording. CHE's next partnership webinar will take place next Tuesday, October 23rd, and is titled Driving the Transition to Safer Chemicals Through Consumer and Retailer Advocacy. To learn more and to RSVP, please visit our website at healthandenvironment.org. CHE's next webinar from the CHE EDC Strategies Partnership will take place Wednesday, November 14th, and will feature the latest research from Dr. Jean-Baptiste Fini. Details will be available on our website soon. If you are new to CHE and would like to stay updated about upcoming events and more, please sign up to receive our newsletter by selecting the Join Us tab at the top of any page on our website. Additionally, if you appreciate these CHE partnership webinars, bringing you the latest environmental health research for free, we encourage you to support CHE's ongoing work by making a tax-deductible donation via our secure website. Again, our website is healthandenvironment.org. With that, I would like to thank Joe Braun for taking the time to talk with us today and Carol for excellent moderation. Thank you so much for joining us, and have a great day.